We're going to begin with our first presenter. It's Julie Etienne. She's from France, Inserm, and she is an engineer in bioinformatics. She graduated from INSA in Lyon, France. She's currently a PhD student in the Cardiometabolic Diabetes and Nutrition Laboratory in France. So, and the title of her talk is Uptake of Fatty Acids by Enterocyte, New Insights Gained from Mathematical Modeling. And Julie's co-authors are Amelie Penhote, Maria Carolyn Mikulski, and Kara Nibble. So we'd like to introduce Julie to present her 5-minute presentation.
Hello, everyone. I'm very happy to be here to present my ePoster at this AOCS meeting. My ePoster deals with the fat contained in our meals. And this fat is mostly composed of triglycerides, which carry fatty acids. In more details, my ePoster focuses on how these dietary fatty acids are being taken up by cells of our intestine, namely enterocyte. In the literature, this process of uptake is very debated, and this process of uptake is also usually reviewed as being composed of passive diffusion along with a carrier-mediated mechanism. We decided to question again this uptake of fatty acid by the enterocyte by asking ourselves if the observed uptake curves were compatible with the pathways suggested by the literature. In order to answer this question, we use mathematical modeling and data fitting at the interface between computer science, biology, and mathematics. And we wrote 3 models of uptake. So first, model M0 considers an uptake only by passive diffusion. Then model M1 considers an uptake both by passive diffusion and a carrier-mediated mechanism. So this is the model suggested by the literature, model M1, and moreover, the carrier E is not clearly identified yet in the literature nowadays. And finally, model M2 considers an uptake by passive diffusion, and this uptake is being followed by an enzymatic transformation of fatty acid inside the cells. And this we know occurs because fatty acids are being transformed to be reesterified into triglycerides. So what did we, to make this a bit clearer, we could imagine fatty acids as being baby sea turtles getting out of their eggs, and we wonder how they cross the beach in order to reach the sea and find other sea turtles. So do they cross the beach alone? This will be model M0. Or do baby sea turtles cross the beach alone, but some are using some kind of carrier like a seagull, like in the movie A Turtle Sail? Or do babies cross the beach alone, but they are somehow encouraged by adults waiting for them at the end of the beach? And once they reach the sea, they feel part of the group, and it's somehow like motivating. So now what did we do with those 3 models? We studied them and we compared them to experimental data extracted from research papers. So as a reminder, I recall the 3 models on the left-hand side of my presentation. And now let's have a look at our results. So looking at the graphic on the top, we can see model M0, where uptake is only by passive diffusion, and we can see that this model hardly reaches the experimental data or stays in the lower part of it. And moreover, if we play around with the parameters, we see that volumes have more impact than permeability. Then in the middle, we can see model M1 and M2, and we can see that both models equally fit the experimental data using nonlinear regression. And finally, at the bottom, the graphic shows the maximal uptake of our 3 models, and model M2 has a greater uptake potential than model M1, with, as a reminder, model M1 being the model suggested by the literature. So as a conclusion, what can we say about the uptake of fatty acids by the enterocytes? Well, first, the pathway suggested by the literature, so here model M1, is compatible with the experimental data, So this is actually reassuring, even if it does not answer the debate. And another interesting point is that other pathways are also compatible with the data. For example, here model M2, that suggests, here model M2 suggests that maybe it is more efficient for the cell to invest in enzymatic transformation of fatty acid inside a cell than into membrane carrier. And last but not least, if our mathematical modeling provides new insight into this uptake of fatty acid by the enterocyte, it also highlights the need of new experimental data in order to quantify and differentiate different uptake fluxes, if more than one. And we hope this step is also the first step of the idea of modeling a full virtual enterocyte. Thank you for your attention, and I am ready for questions.
Hey, thanks, Julie. That was a great presentation. I really like the comparison. Absorption of fatty acids is incredibly important, and there's a lot of malabsorption problems in our world. So it's really a neat model that you have. So right now we're going to talk about questions, I guess, from the judges for you. And we've got our three judges, and they're going to ask some questions.
So I can start. Thank you, Julie. Great work. Well done. Well presented too. I would like that you elaborate on the limitation of this type of modeling.
Well, I guess first is all the hypotheses that we make to model, because it's a model, it's not the reality, and it's hard to know what really happens inside a human or living organism. So I guess this is one of the limitations, maybe the biggest, I guess.
And in terms of error, what type of error is associated to your points, let's say?
Well, first is sorry.
The variation. What is the variability of your curves?
Well, we did not look in the variability. It's more how for example, when we fit the models to the experimental data, how well it fits to the data. So it's the error of the fitting. But in terms of variability, we have, for example, the variability on the parameters we fixed from the literature, or the ones we inferred from the modeling. And for example, in the literature, there are many values of parameters, and we just chose one, but we didn't take into account the standard deviation, for example. So this will be an improvement for our modeling.
Okay, thanks a lot.
Thank you.
Hello, hello, Julie. Great presentation. You said you sort of ground truth this against the literature. Have you got any plans to test this in vivo and in vitro, and how would you go about that?
This is a good question, because in my lab, we work also on Keiko2 cells, so cell mimicking the enterocyte. And the idea is would be to be able to differentiate which fatty acids are being taken up by passive diffusion and which are being taken up by transporters, if any. And I guess this is a wide question, because in the literature it was quite a lot studied before, like in 10 years ago maybe. And I guess it's hard to like get out every genes that could be involved in this uptake, because we don't really know how many and which, and then we don't, I guess, I don't know if we know how to cut off the passive diffusion, for example. This would be an interesting idea, or I know they also talk about endocytosis. So this is another mechanism that maybe involves proteins. So I guess there is many lines of research for experimental experiments.
Thanks, Julie.
Nice job, Julie. Thanks for your presentation. So are there plans in your lab to follow up any of these bioinformatic work that you've done, or?
Yes, it will be great to have new data points, for example, to have to improve our modeling.
Okay.
It's still a topic of discussion inside the team.
Okay. I like the fact that you've used mathematical modeling. It's something very different in this field to assess this. So thanks for that contribution.
Thank you.
Okay. Yeah, thank you. I think putting a model on something really allows you to look at lots of different experimental variables and start to define things. So, neat research. Thank you very much, Julie.